And so I kept telling you that Mendel really had no idea just how complicated everything was. And so we're going to see today just how complicated things can be. So we're going to look at some chromosome abnormalities and chromosome problems. We're also going to talk about sex determination in humans and other animals. So in the 1900s, or early 1900s, biology finally caught up to Gregor Mendel. So there were a variety of scientists who basically rediscovered what Mendel had done, and they were able to study genetics with a little bit more scientific accuracy, not too much more in 1900, but a little bit more than 1850. But there was some resistance that still remained about Mendel's laws of segregation and independent assortment. And of course, we know now today that not everything follows exactly those patterns. So often we cannot predict the end result as clearly as what Mendel did with his pea plants. But still, Mendel's discoveries form the basis of modern genetics. And of course, now we know all about DNA and chromosomes and all those heritable factors, which are actually genes. So cytologists, which are cell biologists, and geneticists began to see parallels between the behavior of chromosomes and the behavior of Mendel's factors. Because by this time we had some pretty good microscopes, and they could see cells undergoing mitosis, and they saw those thick, condensed chromosomes in the nucleus. So they figured there must be something up with those chromosomes. And so, of course, we now know that chromosomes and genes are both present in pairs in diploid cells. Remember, half the chromosomes came from the maternal parent, and the other half came from the paternal parent. Homologous chromosomes separate, and the alleles segregate during meiosis. And so the gametes are haploid, right? The eggs and sperm are haploid. And then when fertilization occurs, the diploid state is reconstituted, right? So a haploid egg and a haploid sperm come together, you form a diploid zygote. So again, in the early 1900s, scientists began to develop the chromosome theory of inheritance. The idea that all those heritable factors are on these chromosomes, and they get passed on from parent to offspring, and that's the basis of inheritance. Thomas Hunt Morgan was the first to associate a specific gene with a specific chromosome. He did this in the early 20th century using fruit flies. And fruit flies have been a model organism for geneticists for a long time, probably now 100 years. Because fruit flies are easy to breed, they reproduce in large numbers, they reach sexual maturity very quickly, and they have some easily observable characteristics like eye color or the wing shape. So there are certain phenotypes that are very easy to study with fruit flies. And so Morgan studied eye color, and he discovered that the white-eyed trait appeared only in males. And Morgan concluded that a fly's eye color was linked to its sex, so a sex-linked trait. So Morgan deduced that the gene with the white-eyed mutation is on the X chromosome. So it's a sex-linked gene or an X-linked gene. And females are XX. They may have two of the red-eyed alleles and have red eyes, or they may be heterozygous with red eyes, right? Because we know that red eyes are dominant to white eyes. Males have only one X, therefore only one allele for eye color. If they have the red eye allele, they'll have red eyes. If they have the white eye allele, they have white eyes. Females can only have white eyes if they have two white-eyed alleles on two X chromosomes. So Morgan was just the first one to associate a specific trait with a specific chromosome. And that was an X-linked trait for eye color. Each chromosome, though, has hundreds or thousands of genes. And the genes are located on the same chromosomes. If they are close together, they are linked, which means they tend to be inherited together, because a crossover event is unlikely to occur in between genes that are very, very close together on a chromosome. So results of crosses with linked genes deviate from those expected according to independent assortment, because the genes are just so close together, they're always inherited together. So this violated the law of independent assortment. The production of offspring with new combinations of traits inherited from two parents is genetic recombination. So we do know that adjacent sister chromatids experience crossover events, right? The arms break off and reattach. We know that homologous chromosomes randomly line up right during metaphase 1 and meiosis 1, so that each daughter cell has a random mix of those chromosomes that came from the original maternal and paternal parents of that individual producing the eggs and sperm. So independent assortment is what helps to give us those random combinations. So every individual is basically a recombinant individual, because we all have chromosomes where the adjacent sister chromatids crossed over and switched places. So we're all recombinant beings in that sense. But linked genes, they move together through meiosis and fertilization. So you can have more than one characteristic that always appears, right? They always appear together. So under normal Mendelian rules, we don't expect linked genes to recombine into assortments of alleles not found in their parents, because the genes are so close together, they're not going to cross over, and you're not going to get those recombination, or those different combinations or recombination events in the gametes. So one of Morgan's students, he used crossing over of linked genes to develop a method for constructing a chromosome map. And this is something that has not been done in a long time. Nobody does linkage maps anymore because we can relatively easily and quickly sequence entire genomes. So there's no need to do this time-consuming and cumbersome technique in order to find the loci of different genes on a chromosome. We can sequence them now. So what we're talking about here is history, how things used to be done. It's not really done anymore today because we have much better and faster techniques. But the idea is that when they created this chromosome map using linkage, they knew the locations of different genes on a particular chromosome. Now, the frequency of recombination reflects the distances between genes on a chromosome. The farther apart those two genes are, the more likely a crossover event will occur in between them. So if the genes are really close together, it's very unlikely that recombination will occur, right? There'll be no crossover event. So by determining the recombination frequencies, how often genes crossed over and switched places that led to a linkage map. Again, we don't do this stuff anymore, but this is just an example of a linkage map from a roundworm genome. And so what you see are the positions of a variety of genes on different chromosomes. And so the idea is if you have a lot of distance in between genes, then it's very likely a crossover event will occur between those genes. And so you get those different combinations. But if genes are really close together, like here for these genes, or here for most of these genes, it's very unlikely that a crossover event will occur. And so you won't get recombination, right? The adjacent sister chromatids are not going to switch pieces. So some genes are linked and they always sort together, right? They do not segregate. They end up in the same gamete. And it's not a problem. There's nothing wrong going on here. It's just that the chromosomes are so close together that they act as one. I mean, the genes are so close together, they act as one. And so they're always found together in the offspring. So the maximum frequency of recombination is 50%. So a 50-50 chance that a recombination event will occur. So that's the maximum value. And if the recombination frequency is very, very low, then those genes are linked together. And so they'll violate the law of independent assortment, right? Because they always migrate together into the same gamete. Now, Mendel, he did study genes which were on the same chromosome, right? The seed color and flower color genes are on the same chromosome, but they're far enough apart that crossing over occurs between them. And so it results in gametes that have the different combinations. So it looks like they're on separate chromosomes. It looks like independent assortment is occurring even though they're on the same chromosome. It's just that crossing over right between the adjacent sister chromatids allows for the different alleles to be switched. And so they act like they're on separate chromosomes, even though they're on the same chromosome. Now, plant height and pod shape, right, the P shape, would show linkage, so they would have violated the law of independent assortments, but Mendel never reported those results. That doesn't necessarily mean that he never did those crosses. It just may have been that those results he could not explain, right, because they violated his law of independent assortment. And so since he couldn't explain it, he just ignored it. So, sex chromosomes. So, of course, there are two primary sexes, male and female. And there are typically obvious differences, phenotypic differences, anatomical differences between males and females. And when it comes to developing, what type of sex, it's not all X's and Y's. It is for humans, but for other animals, they use different systems. But we're going to focus mostly on humans. Our sex chromosomes are the X and Y chromosomes. We know that females are XX, males are XY. But that is not the case for everything. There are different systems, right, for determining sex. There's the XO system, where if you have an X, you're one sex. If you don't have an X, you're no sex, or you're the other sex, rather. We have the ZW system, the haplodiploid system, and the temperature-dependent sex determination system, which we have a slide on that I'll mention because it's so interesting. Where temperature during the incubation of the eggs is what determines whether the eggs develop into males or into females. And it's not the same for every species that does this way. It's mostly in reptiles and some fish, but whether it's warm temperatures favoring males or warm temperatures favoring females, it varies depending on the species. But so humans are XX and XY, right, for females and males. Many insects have the XO system. If they have an X, only one X, they're males. If they have two X's, they are females. So there's no Y here. It's just if you only have one X, you're a male. If you got two X's, you're a female. The ZW system is used in reptiles and birds, because remember, birds evolved from reptiles. And so ZZ, if an individual has two Z chromosomes, they are a male. If they have a Z and a W chromosome, they are a female. So it's kind of the opposite for the XX and XY in humans and many other mammals. And then for some other insects like bees, if they're simply haploid, they're males. If they're diploid, they're females. So a male bee, a drone, only has half the total number of chromosomes. And then females are diploid, right? Because it's the queen bees, the females, who lay the eggs, so they have to go through meiosis. Males don't really do that, right? A male drone doesn't live all that long, right? They're expendable. So just a few words on temperature-dependent sex determination. So this is just FYI, I'm not going to test you on it, but it's really interesting. They've been studying this for a long time and still don't know exactly how it works. But hormones are activated at different temperatures, which leads to the formation of either males or females. The expression of other genes may be activated or deactivated by different temperatures. Usually once sexual differentiation has begun, it doesn't matter if the temperature then changes. That sex that began ends up being that same sex. And sometimes even the genotype doesn't matter. So remember, we just talked about the ZW system, where ZZ is typically male and ZW is female. But it's possible for a ZZ reptile, or like a lizard, can still be female, even though it's genetically supposed to be male, ZZ. And as a female, it can produce offspring. So there are all kinds of weird systems out there. So sex determination is not one size fits all. There are several different systems for creating sex, males and females. So it's not all X and Y, but that's what we are going to focus on in this course, because we're concerned about humans, not all these other animals. But it's just pretty interesting to see that all different kinds of things are possible. When it comes to snakes, they've had some female snakes in captivity which have always been alone. They've never encountered any other snakes, especially no male snakes. And then one day, magically, baby snakes appear. So it is possible for certain animals to reproduce parthenogenetically. So just by themselves, no mate is required, no males, and yet they're able to produce offspring. So all kinds of bizarre things happen in nature. But the XY system, the X chromosome and the Y chromosome are two very different chromosomes. The X chromosome is huge compared to the Y, yet during mitosis, they act like homologous chromosomes. And so they pair up, form a tetrad. No crossing over occurs because the Y chromosome doesn't have any genes that complement the X chromosome, and vice versa. So They do pair up during meiosis and act like they are homologous, even though they're not, right? Because they're very different chromosomes. So when it comes to the formation of eggs and sperm, the X's and Y's will segregate so that the egg has an X, sperm can either have an X or a Y, right? Because in females they're XX, so all the eggs will carry an X. But in males that are XY, half the sperm carry a Y and the other half carry an X. So theoretically, there's a 50-50 chance of having either a boy or a girl. In reality, it's not 50-50. For a long time, it was widely believed that more males were conceived, but more females were born, because for some reason the male embryos and fetuses did not survive to term. More recent data, like from 2015, suggests the opposite, that actually conception is 50-50, but fewer female fetuses and embryos survive to be born. And so actually a slightly higher percentage of males are born compared to females. Something like 51% to 49%. Why, they don't know. For some reason, if there's a problem with that developing embryo or fetus, then mom can miscarry. And the technical term for miscarriage is abortion, right? That's the medical term. So women do spontaneously abort fetuses or embryos approximately 20% of the time. And that's because if there's a problem with that embryo or fetus, mom's body can often detect that and then spontaneously abort it. Often these abortions happen very early in the first trimester, so many women don't even know they were ever pregnant. It's just that there was a problem. It was detected very early on, and so mom's body just spontaneously aborted that embryo, right, to avoid any serious problems later on. So, but it does appear that roughly 50-50 males and females are conceived, but a few more males actually survive to be born than females. So why, they don't know. Now, all embryos are anatomically female. So even male embryos are anatomically female in the very beginning. They all have a vaginal opening, a vaginal slit. In males, it will fuse together, right? The two sides of the opening will suture together. The primitive, primordial, or generic gonads will develop into ovaries in females, and in males, those generic gonads will descend into the scrotum and differentiate into the testes. So all the sex organs are derived from the same tissue in males and females. It's just that when certain genes are turned on, those tissues differentiate into ovaries in females, into testes in males. Other anatomical differences occur as well for the sex organs, but they're all derived from the same tissue, to start with. Now, what makes a male is an all-important gene on the Y chromosome, the SRY gene. And this is the gene that makes males males. So when it gets turned on, it starts activating other genes and activating hormones, which cause a male to develop. So the Y is small, but it's all powerful, right? That one gene, the SRY gene, is what makes males males. Now, the X chromosome has genes involved in sex determination, as well as many other genes, because the X chromosome is huge. So there are many other genes on the X chromosome that have nothing to do with sex. But it does affect inheritance and phenotypes in males and females, because females have two X's, males have one. So if you have an X-linked characteristic, an X-linked trait, the situation is very different and can be very bad for males. So if there is a recessive condition, whether it's, a new, sort of like just a different phenotype or a serious genetic disorder, if it's recessive and it's on the X, males are screwed if they inherit that X, because males only have one X, right? There are no complementary genes on the Y. So whatever genes are on the X, that's what the male is stuck with. But females, because they have two X's, they could have, good alleles on one X and bad alleles on the other X, and they're heterozygous, but they're okay, right? Because they have the dominant allele that's still good and the recessive allele that's bad. But males, if they're unlucky enough to get that recessive allele on the X, they're stuck, right? That's all they got. So females can be heterozygous. Males are hemizygous because they only have one X, so they're hemizygous when it comes to the X chromosome and all those characteristics that may be on the X. So females, even if they inherit a bad X from mom, they can get a good X from dad, and they're okay. But males always get the X from mom, so whatever X they get, they're stuck with. So Duchenne muscular dystrophy is an example of an X-linked trait. And this particular form of muscular dystrophy is the worst one. And so whenever you see these numbers, 1 out of whatever males or females that get affected by this, those numbers are not to be taken as fact, because it depends on what source you look at, what year you're looking at, what country you're looking at. So 1 in 3,500 to 5,000 is just a very approximate number, right? So don't memorize those numbers. They don't really mean much. But it does affect males, because if they get stuck with that Y, which has a defective allele on it for a muscle protein called dystrophin, they get this severe form of muscular dystrophy. And so their muscles are very weak and they deteriorate with time. In this case, those males will only live to their early 20s before they die. There are other types of muscular dystrophy where you can live into your 50s before you die, but it's still not a good prognosis, right? Knowing that you have this. And if you have the form that manifests itself later, you may have already had kids and passed on that characteristic. And so these kinds of things do happen. And in this case, the Duchenne muscular dystrophy is an X-linked trait. So if the males inherit that X with this defective allele on it, then they have this disease and nothing can be done. But women can be carriers, right? They're heterozygous. One good X, one bad X. So they're phenotypically normal, right? Because they're making enough of the protein dystrophin so that they're normal, right? Their muscles are normal. But males have no option. If they get that X, that's all they got. Hemophilia is another X-linked characteristic. It is recessive, so women can be heterozygotes for this condition and be phenotypically normal, right? Their blood clots normally. But males, if they inherit the defective X or the X with the defective gene on it, then their blood cannot clot. And I mentioned this before. This condition was especially prominent in royal families because of all the inbreeding, but it mostly affected males, right? Because if they were unlucky enough to get that bad X, right, then they had hemophilia. And so their blood was very hard to stop the bleeding. These days, we can treat hemophiliacs with clotting factors. So, when people donate blood, sometimes that blood is used to extract the clotting factor from the blood. And that's what is used by hemophiliacs, so their blood will clot if they get injured. But this is an excellent characteristic. It's recessive. Males are disproportionately affected by it because they only get one X, right? They're hemizygous. But females can be heterozygous and they're phenotypically normal, so they're healthy, they're okay. Now, even though females do have two X's, only one of the X's is active. So only one of the X chromosomes, its genes are going to be transcribed and translated, and the other X chromosome is going to be put into a dormant state. And so we call this dormant state a bar body. You can see the bar body shown here in these cells. And so that way in women, only one X is actually active and the other one is dormant. So women are actually a mosaic of their X's because not all cells will have the same X that goes dormant. Approximately half of a woman's cells have one X that's active and the other is dormant. And in the other half of her cells, it's the reverse situation. But still, if she has a heterozygous genotype for a genetic disorder, she has enough of the X's that are the good X's that are active in order to make whatever proteins or enzymes that are needed for her to have a normal phenotype. So even if she has some cells where the defective X is the one that's active and the good X is dormant, that's only about half of her cells. And the other half have the good X that's active. And so she's able to make enough of whatever protein or enzyme is needed so she's phenotypically normal. Men just have that one X that's always active. So Mary Lyon is a British scientist who demonstrated that the bar bodies form. It's random as to which X gets turned into a dormant bar body. And so, like I said, half of her cells will have one of the X's that's dormant, and the other half will have the other X that's dormant. So women, all their cells are actually a mosaic when it comes to their X chromosomes. Half of them have one X that's active, and the other half have the other X that's active. So this can create some interesting situations. There is a defective allele that prevents the development of sweat glands, and it's on the X chromosome. So if women are heterozygous for this allele, they have an X that has the normal allele to make sweat glands, and the other X has the defective allele, which cannot produce sweat glands. And so their bodies are a mosaic, where some cells produce the sweat glands and other cells don't, or the skin, I should say. Right? There are patches of skin which will sweat, other patches of skin that will not sweat. I'll just mention this here because it's really fascinating. that there's also another type of mosaicism, a mosaic where you have two fertilized eggs. So a woman, she gets pregnant, she's got two eggs, and they're both fertilized, but instead of each egg developing into a separate individual, so what we would call fraternal twins, the two fertilized eggs merge together. And they produce one individual who has a mixture of cells which have a very different genetic makeup, right? So one person is actually twins, right? Because their cells have a mixture. Some cells are from that one egg, which was fertilized by one sperm, and the other cells came from a different egg fertilized by a different sperm. Remember, the eggs and sperm are all unique. All those random combinations of chromosomes and alleles. And so this person is a mosaic of those two different eggs that were fertilized. And it's really bizarre. And there was a case where a woman, she gave birth, she's still able to have kids normally. And then the hospital was doing some testing, and they discovered, or they thought they had discovered, that this baby did not belong to this woman. Because when they did the genetic testing, they found different genes, right? When they did something like a DNA fingerprint, two different fingerprints. And they were like, this baby doesn't belong to you. It doesn't have your genes. And so they thought that maybe this woman had stolen the baby from someone else. And it was a huge controversy and required a lot of scientific research to discover that this woman was a mixture of two eggs that had been fertilized and fused together. And so when they took a sample, like a blood sample, to use the DNA from that to do the testing, it just so happened that the blood cells were derived from another egg. And the egg that was used to produce her own eggs came from the other egg that was fertilized, right? Because she has two different cells, types of cells in her body, one from each fertilized egg. And so the baby was produced from gametes that were made from cells that came from that one fertilized egg. But the blood cells came from the other fertilized egg that was part of this woman. And so the genes didn't match. And it took them a while to discover that this woman was a mixture of two fertilized eggs. And the baby was hers. It's just that the baby came from gametes made from one fertilized egg, and the cells they used to test from this woman came from the other fertilized egg. So really bizarre situation where, I am my own twin kind of thing. So all kinds of weird things can and do happen. And we're going to see some more of those today. So let's look at some of them, big problems that can happen that involve the chromosome. So X-linked traits, they deviate, right, from Mendel's rules, right, because you have X-linked genes and there's no comparable gene on the Y chromosome. There are mutations that we talked about, single base changes, but here we're going to talk about whole pieces of chromosomes that get messed up. And so there are major chromosomal aberrations, and their consequences are usually very severe. Often those embryos will not survive, but sometimes they do. And sometimes the consequences are not all that bad. And we'll see some examples of those. So if you have any of these chromosome abnormalities, they're not going to follow Mendel's laws, right? They're not going to follow the law of independent assortment or segregation because the chromosomes are getting mixed up. And so the phenotype is not what you expect it to be in the offspring. So non-disjunction is one of these major chromosome abnormalities that can occur where the homologous pairs do not separate during meiosis 1. So remember, during meiosis 1, all the homologous pairs line up at metaphase, and they're randomly distributed into the daughter cells. But sometimes a homologous pair, the whole pair moves into one of the daughter cells, meaning that that daughter cell now has an extra chromosome, and the other daughter cell is missing that extra chromosome. And so that's a non-disjunction, which will produce gametes, which either have too many chromosomes or not enough. So that's going to be bad, right? Most likely that, zygote, that embryo, is not going to survive because there's just either an extra chromosome or one that's missing. Another situation can happen in meiosis 2, where the sister chromatids normally split and end up in different daughter cells, but one of the pairs of sister chromatids does not split and ends up in a gamete, also producing a gamete that has an extra chromosome, and there'll be one that's missing a chromosome. And so again, if these gametes are the ones that are fertilized or used to fertilize an egg, that zygote or that embryo is not going to survive. So we're talking about a case where the chromosomes, either the homologous pair doesn't separate or the sister chromatids do not separate. So non-disjunctions are almost always very bad, usually lethal. But there's one case we'll see where you can survive with it. You're not normal, but you can survive. Now, we know that haploid means half the number of chromosomes, or N, and diploid means twice the number of chromosomes, or 2N. Well, if you have the wrong number of chromosomes, they use the word aneuploid. So aneuploid, or a state of aneuploidy, means that you're either missing a chromosome or you have an extra chromosome, but you have the wrong number of chromosomes. A trisomy condition occurs where you have an extra chromosome. So you're 2N plus 1. One of the chromosomes, right, you have your normal homologous pair, one from mom, one from dad, and then an extra one, so you have 3. And that's a trisomy. So it's 2N plus 1. If you're missing a chromosome, then you have a monosomy. So you're 2N minus 1. So a person with a trisomy has 47 chromosomes. A person with a monosomy has 45 chromosomes, right? Because normally we have 46 total. Usually, well, if you're missing a chromosome, that's most likely going to be fatal. Most of the time, if you have an extra chromosome, that's also going to be fatal. But sometimes you can survive with an extra chromosome, depending upon which extra chromosome it is. And we'll see some examples. So, of the statement here. If the organism survives, right, with their aneuploidy condition, there will be a distinct phenotype, assuming that organism survives. Usually it does not. But if it does, you know there's something wrong. So aneuploidy can occur because of failures of the mitotic spindle. So aneuploidy can happen early in development. Then it will be passed on by mitosis, right, to a large number of cells. So everything may have started off okay, but then a mistake happened, right, during mitosis and cell division. So all the cells produced at that mistake are all messed up. And yes, it will have a substantial effect on the organism, usually killing that organism. Now, some organisms naturally have multiple sets of chromosomes. So instead of 2n, they could be 3n or 4n, right? They have multiple sets of chromosomes. We call multiple sets of chromosomes polyploidy. Remember, diploid is 2n, a triploid would be 3n, a tetraploid is 4n. And there are organisms like plants, many plants are tetraploid, right? It's normal to have 4 sets of chromosomes, and that usually makes those plants larger, more vigorous, stronger plants. That doesn't really work for most animals. Having extra sets of chromosomes is not a normal condition, with some exceptions.
How do you
Yeah, question?
any question? Only one very smart guy in class. One this guy. One being to back into the question. Say one thing I learned when I was in my GED and all that. One thing I'm not helping in English.
I can't hear you, Dana Lola.
I always talk. I just keep quiet like this.
Yeah.
You know, so just all the notes here. What's me so it was to the point that the guy, teacher mentioned only those that always keep the thank you to those that always talk in class that the smartest.
You're breaking up a lot, so I can't hear you. So again, many plants are polyploid, and that's normal. Very few animals are polyploid. There are some fish and certain amphibians that have polyploid species, but when it comes to most mammals, it's very rare. There may be a rodent which is polyploid, but for most animals, having extra sets of chromosomes is a bad thing and just doesn't work well. But in those organisms that do survive or are naturally polyploid, their phenotype is basically normal. If you have just one extra chromosome or you're missing a chromosome, you're usually not normal, and you can see that. So one extra or missing chromosome upsets the genetic balance during development, and so it produces more severe consequences, right? If you have extra whole sets of chromosomes, again, depending on the organism, that may work out just fine. Now remember that during crossing over, the adjacent sister chromatids break and they reattach. Sometimes things happen, right, where maybe when the chromosome breaks, it floats away, it doesn't reattach, and you have a deletion. So not just one gene, but a region of the chromosome with many genes gets deleted. So that's normally going to be very bad. A duplication occurs when a fragment gets attached as an extra segment to a sister chromatid. So one sister chromatid is missing a piece, but the other one has the extra piece attached to it. This is going to cause problems when it comes to gamete formation. So those individuals, if they survive, are often sterile because they can't produce gametes correctly because their chromosomes are messed up. Right? So they still have all the genes. It's just that when it comes to making gametes, it just can't happen correctly. Inversions occur when the chromosomes break off and reattach, but they, turn around, they reverse and reattach. So we see that here. Normally the pattern of genes ABCDE is ADCBE. Again, this individual still has all the correct genes. The problem occurs during gamete formation, during meiosis, because now a crossover event is not going to occur in the correct place because the genes are no longer in the correct place on one of the homologous chromosomes. So crossing over does not occur correctly. So you often have problems with fertility or maybe sterile. With a translocation, here, when the adjacent sister chromatids, when the arm breaks off, instead of switching places with the adjacent sister chromatid, it kind of floats away and gets stuck to a non-homologous chromosome, so a different chromosome altogether. And that's a translocation. And again, these individuals still have all the genes they need, and so they can survive, but they're often infertile or sterile, right? Because they can't form gametes correctly. But they still have all the right genes, and so they'll be phenotypically normal. It's just that they may not be able to have children. So, but it depends on what piece of chromosome it is, what genes are on it, where it ends up as to whether or not it'll be a lethal condition or become a serious genetic disorder, or be phenotypically normal but sterile. And there are several serious human disorders which are the result of alterations in the chromosome number or maybe the structure, right? So either a deletion or translocation or an inversion. And in humans, we do have a lot of chromosome abnormalities, right, that occur. And this is one of the reasons why women do spontaneously abort embryos, is because there were serious problems. And so a woman's body can detect many of these problems and spontaneously abort that embryo, right, before it has a chance to develop too much. There are certain other conditions where you have an extra chromosome and you do survive, right, to be born. You're able to grow up to be an adult. Not a normal adult, but you're able to survive. So the best known example of an aneuploid condition where these individuals have an extra chromosome is Down syndrome. And so Down syndrome, also known as trisomy 21, because they have 3 copies of chromosome 21. These individuals survive to be born, and they can grow up to be adults. They are not phenotypically normal. They all have a very similar facial structures, facial characteristics, so you can usually recognize a person with Down syndrome regardless of what race or ethnicity they are, because they all have a very similar appearance. They are developmentally disabled, they're slower, IQs are lower, they may have some mental retardation. They're often very functional human beings, not necessarily being able to live completely independent lives, but they're often very functional human beings. But this is a case where there's an extra chromosome number 21. It does affect, again, about 1 in 700 children, but that number is just a rough approximation, and that comes from US data. So chromosome 21 is a very small chromosome, and that's probably why these individuals are able to survive, because there's not as many genes on that small chromosome as there would be on other chromosomes. So having an extra chromosome number 21, you can survive, but again, you're not phenotypically normal. Now, Down syndrome is the result of a non-disjunction, right, during a gamete production. And this typically occurs in the production of eggs in women. And so the frequency of Down syndrome increases with the age of the mother. So if you're over 40, they often will perform an amniocentesis to remove some fetal cells so that they can be examined to determine if the child has Down syndrome or not, right? Because they can count the number of chromosomes and see. And remember when we were talking about gamete formation and how males start producing sperm at puberty and produce them, daily for the rest of a male's life. But women produce their eggs during fetal development, and the eggs go through meiosis 1 and then stop. At puberty, those eggs start going through meiosis 2. When a woman reaches age 40 and beyond, those eggs have been sitting there, stuck after meiosis 1 for 40 or more years. During that period of time, some bad things can happen. And so when that egg starts to go through meiosis 2, some of the chromosomes may be sticky, pieces may have broken off and floated away, right? Because they're not, the eggs are not in suspended animation, right? They're still living cells. It's just the chromosomes are just sitting there, right, for 40 or more years, and that can lead to problems. There are trisomies of other chromosomes, right, that can also increase with maternal age. But normally with these other chromosomes, if you have an extra one of them, that's a lethal condition. And so that those offspring either are spontaneously aborted early in development, or they die soon after being born. So when it comes to the sex chromosomes, X and Y, there are a variety of non-disjunction conditions, right, that occur in humans, and the humans tend to survive pretty well with extra X's and Y's. So the sex chromosomes don't really mess up the genetic balance like the autosomes do. Remember, the autosomes are chromosomes number 1 through 22, and chromosome 23 are the X and Y's. And so it's just not as bad to have extra X's and Y's as it is to have extras of all the other chromosomes. When it comes to having an extra Y, it's not really much of a problem because there's very few genes on the Y. X's, if you have extra X's, only one X is going to be active, and the other X's will be turned into bar bodies, so they're going to be deactivated. And so that's why the sex chromosomes, having extra of them is not that much of a problem in terms of survival, but you may be infertile, right? You may be sterile because you still have the wrong numbers of sex chromosomes, and that creates problems during meiosis and the formation of gametes. Klinefelter syndrome is a case where a male is XXY. So a male that has two X's. No, these are not gay males. XXY does not affect your sexuality. Because remember, one X is going to be a bar body. So only one X will be active. So basically an XXY male still has only one functional X and then the Y. They do have male sex organs, but as I said, they're sterile because they can't form sperm with the right numbers of chromosomes. And they may have feminine characteristics like enlarged breasts, extra body fat, but their intelligence is normal. And so you really cannot look at a guy and say he's XXY, right? Because if a guy just becomes overweight, guys deposit fat in their breast tissue, right? Just like women do. Now, XYY males are males that have two Y chromosomes. And this doesn't really cause any significant problems. They may have some learning disabilities, some language disabilities. They tend to be taller than average, but Just because a guy is tall, over 6 feet tall, does not mean he's XYY. Now, a long time ago, they used to think that XYY males were more likely to become criminals, right? Because males are more criminal to begin with. Having an extra Y means you're extra male and extra criminal, and that's not true either. So XYY males are basically phenotypically normal, right? You can't tell by looking at a male he's XYY, right? But he may be sterile. And there's other weird things too. You can have XXXY, right? So you can have extra X's and Y's. Right? So there are a variety of different sex chromosome abnormalities. We're not going to go into all of them because those tend to become very rare. Trisomy X is where you have 3 X's. So again, once in every 2,000 births, it's not a number worth remembering, so don't worry about it. The females are healthy because the extra X is going to become a bar body, right? It's going to become inactive. And so they basically look like normal females. They can have some developmental disabilities, may have fertility problems, right? But again, you can't look at a woman and say she's XXX, right? She looks phenotypically normal. Monosomy X or Turner syndrome is where a female just has 1 X and nothing else. Again, not very common, 1 in every 5,000 births. They do look like females, but they're physically immature. They don't really develop fully into a female body. And again, they're going to be sterile because, they can't make gametes correctly. They have a variety of symptoms. We have some other chromosomal abnormalities, which involve some deletions. And so even in a heterozygous state, so again, you have a good X and a bad X, it can still lead to some serious problems. And there's one syndrome, Cri-du-chat. Those of you who speak French know what that means. And what does that mean?
Crying of the cat.
Right, so Cri-du-chat. So basically, these individuals, they're babies and toddlers, they sound like they're meowing, like a cat does. And this is the result of a deletion in chromosome number 5. So they're missing a piece of chromosome number 5. Even if they have a good chromosome number 5, right? A homologous, pair where one's a good chromosome 5 and the other's a bad one, it still causes problems because they're missing a piece. So they're missing those alleles from that chromosome. And so these individuals are very developmentally disabled, small heads, unusual facial features. And like I said, they cry like a cat that's meowing, especially when they're very young. And about 10% of individuals born with Cri-du-chat syndrome die. But the ones who survive will never be normal. They will always be institutionalized or require care throughout their entire lives. But a piece of chromosome number 5 is missing. So even though they have a good intact chromosome number 5, it causes a severe genetic disorder. Chronic myelogenous leukemia is the result of a chromosomal translocation where there is a fragment of chromosome 21 gets stuck to the tip of chromosome number 9. And so this leads to a form of cancer, of blood cancer. When it comes to those individuals that have Down syndrome, sometimes that chromosome number 21 is attached to another chromosome. So they don't just have 3 separate chromosome 21s. 21 is sometimes attached to one of the other chromosomes. So, with the exception of the X-linked characteristics, if you have an autosomal trait, so a gene that's on a chromosome 1 through 22, so those are the autosomes, you would expect that it doesn't make any difference which parent you inherit that, chromosome from, right, that allele from. And most of the time that's true, because when we do our Punnett squares, we don't care which gametes are coming from which parent. The gametes and the genes in those gametes end up in our Punnett square, and we assume that those individuals, those offspring, are going to have whatever phenotypes are encoded by those genotypes. And we don't care whether they're male or female, because normally that makes no difference. But there are some bizarre examples where it does make a difference if you get that allele from mom or you get it from dad. And if it's a genetic disorder, it manifests itself differently. So you have a different phenotype depending on whether you inherited that chromosome from mom or dad. It's really bizarre. So we have 2 disorders, a Prader-Willi syndrome and Angelman syndrome, which are 2 different manifestations of the same chromosomal abnormality. So there is a deletion of a little piece of chromosome 15, but whether you get the abnormal chromosome 15 from mom or from dad, you get a different disorder. So if you inherit that abnormal chromosome from dad, you get Prader-Willi syndrome. It's characterized by mental retardation, obesity, short stature, unusually small hands and feet. But if you inherit that abnormal chromosome 15 from mom, you get Angelman syndrome, which has an entirely different phenotype. Spontaneous laughter, jerky movements, other motor and mental symptoms. So it's the same deletion on the same chromosome, but it manifests itself as 2 different genetic disorders depending upon which parent you inherited that chromosome from. So that is a really bizarre thing. You would not expect that to be the case. Why is it that way? Well, there's something called genomic imprinting. And as we'll see, there are genes that can be methylated. So the cytosine nucleotides can have a methyl group attached to them. And normally methylated genes are turned off. They're not transcribed or translated. And so it depends on which chromosome had been imprinted, which one had the methylated genes, so whether or not it came from mom or dad. Now, if you have cells that have a genomic imprinting, when you go to make gametes, when meiosis occurs, the imprinting disappears so that gamete formation can occur normally, but after the gametes are formed, then the genomic imprinting reappears in exactly the same way it was before the gametes were produced. So, a very bizarre condition, but one that's methylation is apparently a relatively common thing that does occur with many of our genes and is actually responsible for normal development. But the idea, if you methylate the cytosines in a piece of DNA, you silence that DNA. It's not going to be transcribed. And so no proteins will be translated from it. This kind of modification is called an epigenetic modification. So it's epigenetic. You still have that same gene, you still have that same allele, but it may be turned off. So let's say you have a dominant allele and a recessive allele. Normally, you would expect to see the dominant phenotype. But if that dominant allele is methylated, it's turned off. And so what you see is the recessive phenotype, even though you have the dominant allele. So once again, it is way more complicated than Mendel, right? Mendel's pea plants. So way more complicated. You can have a gene, not see the phenotypic expression of that gene because of things like epistasis that we talked about, where one gene is modifying the phenotypic expression of another gene. You can have cases of pleiotropy where you have multiple genes and multiple alleles interacting together, right, to produce unique phenotypes. We have the norm of reaction where the environmental conditions can affect the phenotype, even though two organisms may have the exact same genotype, their phenotype is different because of the environmental conditions. And here we see yet another way to modify gene expression with an epigenetic modification like DNA methylation. So you can have an allele, it's not expressed because it's methylated, it's turned off. So very unusual things happen when it comes to genetics. And there's still a whole lot we do not understand. And even more bizarrely, you can have a gene that's not methylated, but not being methylated can result in that gene being turned off. So it's very confusing. But in general, we talk about DNA methylation as a way to turn off a gene. But, many different things happen, right? In bacterial cells, their chromosome gets methylated as a way to protect it from restriction enzymes, right? Because restriction enzymes, we know, cut up DNA. Why don't they cut up the bacterial chromosome? Because it's methylated. But those genes are still transcribed and translated. It's just that methylation protects them from the restriction enzymes. But an invading viral DNA is not methylated and can be destroyed by the restriction enzymes. So methylation is hard to explain because it manifests itself differently. It may silence genes, it may not silence genes. It protects genes from destruction in the case of bacterial cells, but it's an epigenetic modification. So it's not a different allele. It's the same allele, but it's methylated, and that may or may not turn it off. It usually turns it off. We have many genes which are methylated and turned off. And this is actually important for normal development. So again, just having a particular gene, a particular allele does not necessarily mean you're going to express that characteristic. It's very complicated. Fragile X syndrome. The X chromosome is fragile, meaning it can break. And this usually happens at the tip, where the tip of the chromosome is just held onto the rest of the chromosome by one, strand of DNA. And so it can break off very easily. If that happens, those genes are lost. And so it's normally not a good thing. And it can lead to different degrees of mental retardation. And it appears to be subject to genomic imprinting. And males are disproportionately affected by this condition because males are hemizygous. They only have one X. If they inherit the fragile X from mom, like I said, they're screwed. There's nothing complementary on the Y chromosome. But females can have a normal X and a fragile X. Half of the cells, the fragile X will form a bar body and be dormant. And the other half of cells, the fragile X will be the active one, but they got enough, good cells. And so there will still be problems, especially if women are homozygous for the fragile X, but it's not anywhere near as common in women as it is in men. And imprinting by the mother somehow causes it. So somehow methylating genes on the X causes it to become fragile, but they don't really understand why. So that's what happens with the autosomes 1 through 22 and the sex chromosomes X and Y. But as you know, not all the DNA in a cell is in the nucleus. Mitochondria and chloroplasts have their own chromosome, right? They have their own genes, and they can have their own problems. So mitochondria and chloroplasts do not exhibit Mendelian genetics, right? There's no independent assortment or segregation with a circular chromosome. It doesn't work that way. Now, we know that during gamete formation, sperm only contain chromosomes, right, from dad. The egg cell contains the chromosomes from mom, as well as all the mitochondria. So mitochondria are maternally inherited. So all the mitochondria we have came from our mothers, which came from their mothers, and so on and so forth. So all mitochondria are maternally inherited. So it's theoretically possible to analyze mitochondrial DNA to determine evolutionary relationships, including ancient maternal ancestors. There are defects in mitochondria. People do suffer from mitochondrial diseases. Usually they fatigue very quickly because they're not able to make as much ATP. Sometimes they just have far fewer mitochondria than they should. During egg formation, something happened and all the mitochondria weren't there. And so they're not able to make as many new mitochondria. And in other cases, the electron transport chain is disrupted. And so the electron transport in chemiosmosis, right, to make ATP, it just doesn't work well. And this could be a genetic condition. It could also be caused by certain heavy metals. So heavy metals are poisons, and they can interfere with electron transports. And so you can develop a mitochondrial disease if you're exposed to a lot of heavy metals. I forget who it was, but there was some guy, I think it was a former cyclist, not what's his face, not the guy that used all the performance enhancement, not Lance Armstrong, but someone before him, a famous cyclist. And he accidentally shot himself or got shot with shotgun pellets. And shotgun pellets are made from lead and other heavy metals. And so they weren't able to remove all the shotgun pellets. And so they poisoned his mitochondria. And so he suffered from severe fatigue for a long time. There are certain drugs that can help, but mitochondrial diseases typically result in fatigue, right? Because you are not able to enough. Oh, Greg LeMond, is he the one? So all kinds of unusual things can happen. And there also was a baseball player. I can't think of his name. I think he's a coach now. When he was young, in his 20s, he started having severe fatigue. He just wasn't able to play baseball well, and they discovered he had a mitochondrial disease. And so he had to, leave baseball. He couldn't play, right? Because he just didn't have the energy to play, right? He didn't have the stamina to play. But now I think he's a coach, maybe a manager, but I think he's still associated with baseball. It's just that he can't play because he just doesn't have the energy. He's not able to do it. So mitochondrial diseases typically affect ATP production, so you suffer from fatigue. and they can contribute to other things like diabetes, heart disease, other diseases of aging. So we have a lot of DNA in our cells. Most of it's in the nucleus, but some of it's in our mitochondria. And we have mutations and defects, right? None of us is genetically perfect, right? We all have, DNA problems, most of which are not that significant, but no one is genetically perfect, right? We have a lot of accumulated mutations and defects and all kinds of things. Questions or comments? I know it was a lot and I didn't stop, but right, so genetics is complicated. So if you're interested in becoming a research scientist, genetics is a very interesting field of study. Right? There's a whole lot of stuff still to do when it comes to genetics. Remember, the genome is all the DNA that's in a cell, all of it. And when it comes to your chromosomes, remember, a lot of your DNA does not code for proteins. It codes for RNAs, which can regulate gene expression. So next week, we'll have our last lecture on the regulation of gene expression, which I think is on Tuesday. Monday, we're going to be playing with genetics problems. So you should play with those genetics problems this weekend because there's not enough time for me to go through everything in the class. So you need to start playing with them, right, to see how to do those Punnett squares, right, how to determine genotypes and phenotypes. When it asks you for the frequencies of a genotype or phenotype, you can express it differently. You can express it like a 3-to-1 ratio. You can say 75% this or 25% that. You can say 0.75 and 0.25, right? So there are different ways to express those numbers. And I don't care which way you use. Right, but definitely take a look at those problems under the course content for today and start looking at them and try doing some of them because you will see some of them on exam 4. So Monday we're just going to do genetics problems. So questions, comments?